So in one of the lectures for the second day, we're going to talk about cell changes, cell alterations. Now that can be good. If you lift weights, your cells get bigger and you get stronger. Okay, that's the response to eustress, EU meaning good, eustress. You have, if you go running, your cells get stronger, your mitochondria get better, and you use oxygen better. And so that's a cellular adaptation that allows you to do things better than you did before. And that's a great thing about the human body, is that most of our cells, if you put a stressor on them, they will adapt and get better if it's a reasonable adaptation. But since this is pathophysiology, we're also going to mainly look at the changes that happen when something goes wrong. Cells will try to adapt, and they'll either succeed or they'll die, or they'll change into something else like a cancer cell or you know, a inflamed cell, and therefore the adaptation won't be a successful one. And that's what we're more interested in for pathophysiology. So here are some of the things that will happen to cells besides cell death, and we'll talk about that too. Atrophy, meaning the cell gets smaller. It could be adaptive versus pathogenic. If you become a couch potato, your cells will get smaller and you'll get weaker. Pathogenic, if you get a disease, the cells can atrophy away. Or if you break your spinal cord and you're paralyzed from the waist down, the cells will atrophy. Hypertrophy, they get bigger. Hyperplasia, you get more of them, the same kind of cells. Metaplasia, you add a whole bunch of new cells. It's usually an appropriate adaptation, maybe to something that's going wrong, but your body is doing the best it can to make it better. Dysplasia is when the cells alter so dramatically that they don't really work right anymore, turning into cancer cells, turning into a tumor. So here we have, if we have normal cells here, atrophy, the cells actually shrink and the internal contents shrink, and they then are not able to do their job as well. Hypertrophy, those same cells get bigger. That can be good or bad, but you know, hypertrophy, those same cells are getting better. Hyperplasia, you get more of them. They don't get bigger, but the tissue increases in size because it has more cells in it now. Metaplasia, you get a slightly different surrounding. Meta means around or beyond. Plasia of growth of cells, for instance, to try to cover a hole in an artery or something. And dysplasia is when the cells are so distorted that they don't work right, such as cancer. Here's what's going on with my brain these days as I age. Here's a young person's decent brain. You have the pia mater over it out here. It's like saran wrap, but you can see that the gyri and the sulci, the gyri are big and the sulci are very skinny. They're smashed together because you have as much tissue here as possible. With age, degeneration, Alzheimer's, the spaces between the gyri get big because the gyri are shrinking. You're actually having an atrophy of the brain. Here we have hypertrophy of myocardial cells, of heart cells. When you get some kinds of heart disease, or let's say you have chronic hypertension and your heart over time becomes muscle bound. If the muscle just gets bigger trying to push against that higher pressure all the time. You don't get more cells, the cells just get bigger and bigger and bigger. So this is a hypertrophy situation of cardiac muscle cells. You can see how thick the chamber has gotten. Here we have normal. Here we have metaplasia. So let's say this is your airways and you had an infection in there and it killed some of the epithelium. You will grow new cells to try to cover that patch, And then hopefully normal epithelial cells will be able to grow over the top of that. So and then this will kind of remodel and get down to a relatively normal size again. That's what you would hope to have if you had an irritation, if you had chronic lung smoking. Initially, you start doing this. It kills some of the cilia, but at least you replace the cells with non-cancerous healthy cells. Dysplasia, the cells have become totally distorted. They're cancerous. You can see here they're showing that they're dividing and dividing and dividing really fast. So this would be the cancer of that same tissue. So when a cell is stressed, it will adapt the best it can. Eustress usually means the cell will get bigger, more active, more enzymes, more mitochondria, whatever it takes for that cell to do its job will increase. You could also split it and have a hyperplasia of that tissue if you need more cells to do the job. If it doesn't adapt sufficiently because it's an injury, a situation that it can't truly adapt to, the cell will become injured. Now it can either reverse that injury, heal itself, and get better and go on living, or if the injury is irreversible, if you don't solve the underlying problem in time, the cell will die. The most common injury mechanisms for cells, well, some of the really common ones involve not enough oxygen. You get a blocked artery. You get a blocked coronary artery, and it first injures and then kills cells downstream if you don't do something about it. Loss of blood flow to your legs. Cutting off your arm and having it sit without blood flow for a while before you put it back on. So you're going to have first an ischemic condition, which means you don't have enough blood flow to that tissue. Oxygen levels are dropping and the cells are starting to malfunction and become injured. Anoxia, when you have no oxygen going, like if you stop breathing, you'll have anoxia to the brain, and a very short amount of time is taken before irreversible injury can happen there. If you cut your arm off, anoxia until you get it put back on and get blood flow restored. And reperfusion injury we'll talk about, because this is a big deal. If I cut my arm off and it goes without oxygen for an hour, and then I put it back on, you can usually get, nowadays they're so good at it, they can get the arm to work again. But when you first reintroduce blood, those damaged tissues are so starved for oxygen and healing that they over-respond to this huge influx of oxygen. They dilate massively to let the maximum amount get through. They let white blood cells bind to the cellular membranes or to the capillary membranes to try to heal stuff. And this overflood of oxidative energy causes reperfusion injury. It causes sometimes more damage than you had in the first place. They have to be really careful with that when they reperfuse a part of the brain. If somebody's had a stroke and they have to dissolve the blood clot, then sometimes that burst of oxygen coming in there causes enough additional cellular damage that is almost worse than the original blockage was. So one of the main things that happens before the cell actually dies from lack of oxygen, what happens is oxygen is required to make ATP. The mitochondria use oxygen to make ATP. You need ATP all the time in all of your cells to run the sodium-potassium pumps on their membranes to keep the sodium outside, the potassium inside. And sodium-potassium pumps pump 3 sodiums out for every 2 potassiums in, so they keep more stuff out than in. If you stop those pumps, the sodium starts building up inside. It increases intracellular osmolarity, and it sucks water into the cell, and it swells up and pops. And that's one of the main things that kills brain cells when you have a stroke. If you don't solve that problem pretty soon, the cells start building up intracellular sodium, they swell up and they start bursting, and once they're gone, they're gone. You don't replace cells like that. So here is anoxia. Okay, you got a thrombus, you have a blood clot, you can't get red blood cells there, no oxygen, the ATP pumps stop working, and therefore sodium builds up in the cell. It starts sucking water in by osmosis, and the cell swells up. And it can kill the cell if you don't correct it pretty quickly. Reperfusion injury is when you suddenly remove that clot, the burst of oxygen does two things. It causes a lot of neutrophils, a lot of your white blood cells to come in and adhere to the vascular membrane, because those cells have sent out all kinds of emergency signals. We're in trouble, we're in trouble. And the neutrophils don't know any better. They think maybe there's an infection. So they all come, start penetrating the walls, and that causes vascular damage. But oxygen bursts, huge amount of oxygen, produces what are called oxygen-free radicals, or reactive oxygen species, which are very harmful to a lot of kinds of tissues. Free radicals, reactive oxygen species, ROS, is the way they're usually listed now, because reactive oxygen species covers a little bit wider range of things. These are either cells or atoms or groups of atoms, maybe a molecule or a clump of atoms that has essentially become oxidized through this burst of oxygen and has now lost or gained an electron on the outer ring, or gained an outer ring with an extra electron, or something, in some manner it's developed on its outer ring an unmatched pair of electrons. That's not what you want, because normally these molecules and atoms that you have in there are stable and don't react with stuff, and so you can have homeostasis with them floating around. But if you produce a situation where that atom has an unpaired electron, it's going to start reacting and binding to stuff, trying to find a second electron to make up the outer ring. And so if it binds to a lipid, it peroxidizes that lipid. If it binds to a protein, it changes the structure of the protein. DNA, if it binds to DNA, it changes the structure of the DNA and can alter it to the point where it produces insufficient or incorrect types of proteins. Causes mitochondrial damage. There's a lot of structures in the mitochondria that use oxygen to make ATPs. And the main thing is, if you take any substance and bind it to a working substance, it changes it so it doesn't work anymore. So that's oxygen-related cell problems. Ischemia, anoxia. Chemical injury happens when the chemical actually reacts with the cell membrane or some structure in the cell, kind of like a reactive oxygen species would, but it somehow binds, reacts, and causes it to malfunction. It kills the cell or causes it to act inappropriately. Things like mercury or lead, they often don't kill the cell, they alter it to the point that it acts so differently that it makes the person crazy, or it makes them go develop essentially birth defects. So lead and mercury have those kind of longer-term effects, especially on fetuses. Carbon tetrachloride is a very common chemical that damages cells. Fire extinguishers, refrigerants, a lot of home cleaners that you might get on your hands. Alcohol, you can metabolize 1 or 2 drinks a day if you don't drink them too close together. But if you exceed that, your metabolic activity in the liver gets overwhelmed. The alcohols build up. They start damaging the hepatocytes in the liver. They start damaging brain cells. They start damaging a lot of different kinds of cells. Any number of drugs, especially things like spice, like ecstasy, cocaine. Some of those drugs will alter cells to the point where they can cause heart attacks. They can cause long-term brain injury. Here's a kid that was born with fetal alcohol syndrome. The mother was drinking to excess. Any drinking is excessive when you're pregnant, but if you drink enough, it does such alteration of the growing cells in the child. They tend to be born with microcephaly, unusually small head, small brain, changes to their eyes, changes to their jaw structure, and mental retardation. And it's irreversible. Here's the liver of an alcoholic. Now this is a vacuole full of some platelets and things, but what they're showing here, this chicken wire fibrosis, is they've stained it so that you can see it. And it's fibrotic tissue, connective tissue, that is now kind of encapsulating these cells, and it's going to decrease the liver's ability to do its job, to filter things out, to make bile. If the liver cells can't properly make bile, the bilirubin will build up in your bloodstream and you'll turn yellow and you'll have jaundice. And another way to cause a cell to malfunction is to smash it, cut it, beat it up, stab it. So a variety of injuries. And now we're going to start getting into the part of the course, well, the rest of the course, where we're going to have all kinds of very graphic photos of things that happen. What this kind of cancer looks like, what this birth defect looks like, or what a stab wound looks like. Because there are a variety of different things that can, or types of injuries that can cause cell death, cell injury. A contusion, bang, big bruise, a lot of bleeding under the skin at first from broken capillaries, turns purple, turns yellow as it starts healing. Okay, tissue damage with bleeding, could be under the skin, could tear the skin and be on the surface too. So that's like a blunt force trauma. A hematoma is a collection of blood. If you hit your head really hard and it swells up this big, I've seen it happen where actually my wife was in a car accident and she hit her head against the steering wheel so hard that she had a hematoma that was the size of a softball on her forehead. And it was just a big blood blister, sort of. Swelling of tissue, but also filled with blood. Oma means tumor, hema means blood, so a blood tumor is the actual term. An abrasion or a scrape, if you fall off your bike and slide across the road for a little while, you're going to get an abrasion. A laceration is a cut. Cut yourself while you're everything from a paper cut to having your throat cut. And a fracture is a break, a broken bone. We'll deal with fractures more when we get to the skeletal system. Blunt force trauma. This wasn't a cut. This person caught their foot in some kind of a machine that was going this way, and it just wiped the skin off. So this would be a blunt force injury. It could be everything from a small bruise to a being hit in the head by a large object that doesn't penetrate but causes maybe even a skull fracture inside. Sharp injuries can happen intentionally or unintentionally. I've seen episodes of TV shows where people fell off the roof and landed on their fence post, and it wound up going in here and coming out their shoulder, and they go into the hospital and have all of this trauma. So it can be accidental, of course, stabbing yourself by accident when you're trying to cut something, stepping on a nail, or intentional, stab wounds, puncture wounds, chopping wounds, slices, trying to slice your wrists because my tests are too hard. A laceration, sliced throat. Obviously, not a good laceration to have. How about gunshot wounds? Fairly common in this country. Entrance wounds, it makes a little tiny hole exactly the diameter of the bullet. They can look at it and usually tell whether it was a 22, a 38, or a 45, or a 357 Magnum. The entrance wound going in just pokes a hole. The exit wound is called a shored exit wound because as it comes out, it kind of blows the tissue away on its way out. Entrance wounds, it depends how close you are. If you're real close, the skin will be burned from the exploding powder. Intermediate range, it gets what's called stippling, where some of those powder fragments just hit the skin and made little tiny holes or burned dents around the gunshot wound. And then if you're any farther away, there's nothing but the hole. So they can have a pretty good idea how far away the person was. Here's an intermediate range with a hole with stippling around it. And here are a couple of exit wounds. And you can see that the hole is many times the size of the bullet as it blew the skin apart on the way out. Other types of injuries which can happen, again, either intentionally or not: suffocation, strangulation, asphyxiation, or drowning will provide you with an overall body anoxia and cause cell death, first of tissues that are most important like your brain and heart, and then cell death of other cells too if it goes on long enough. Suffocation means that you simply, for one reason or another, didn't stop getting enough oxygen. If you're locked in an old refrigerator and you use up all the oxygen, you suffocate. A baby might suffocate if it rolls over on its face, burying its face in its pillow so that it can't breathe. Or you could have a bad guy come into your house and take a pillow and put it over your head like they do in the movies. Or plastic bag over your head. Strangulation means cutting it off at the throat. It can be intentional or unintentional hanging, manual strangulation, what you like to do to your professor when their tests are too hard. Chemical asphyxiants. Cyanide blocks your oxidative processes inside the cells. It doesn't keep you from breathing in the first place. It causes the cells not to be able to utilize oxygen. Other things you might breathe in cause an anaphylactic reaction that closes off your airways. And so you're unable to move air through the airways because that chemical has caused an asphyxiation. Cyanide does that. Carbon monoxide, in a way, is a chemical asphyxiant. It locks up all of your hemoglobin so it can't carry oxygen. Or drowning. We'll spend more time in this course with diseases. That's what pathophysiology is. Now, a pathologist certainly looks at gunshot wounds and stab wounds, but also we're going to be looking at different kinds of diseases caused by invasion and destruction by bacteria, fungi, viruses. The pathogenicity of a substance is its ability to actually cause disease. It might be something that it can enter you and doesn't do much. On the other hand, if it multiplies inside and causes a disease directly, or if it causes a hypersensitivity reaction, an allergic reaction that causes you to die because your own body is fighting it off in a way that's harmful to you. We're going to have a whole lecture just on infection. So we'll talk a lot about bacteria, viruses, and fungal infections. That's just one of the ways that you can have injury to cells. Your immunological and inflammatory response is a really big deal. It's designed to save us. If it gets out of control, it'll kill us. So it depends on how you respond to a pathogen as to whether your own immune response will make you sick for a little while but then get better, or whether your own immune system goes out of control and winds up killing you. First of all, the white blood cells will come. They will eat the bad guys if they can, if they recognize and target them well enough. But at the same time, they'll have a tendency to release signaling molecules, cytokines, to turn on other processes so that your immune system relates one part to the other to bring the entire immune response into play. Phagocytic cells, mainly our neutrophils or white blood cells, like to eat the bacteria or viruses. Mainly bacteria. They're really good at that. And generally speaking, a white blood cell will eat one bacterium, kill that cell, and then die in the process. It's like a honeybee, can only sting one time and then it dies. So they are, sacrificing themselves to save your life. And white blood cells, accumulating dead white blood cells, are pus. That's what pus is. Immune and inflammatory substances, our inflammation and our immune systems, lectures will talk about those. Again, designed to activate other parts of the immune system. And then membrane alterations can happen when your own immune cells stick to the side of your vascular walls and penetrate to access the bacteria that are in the tissue. Your bacteria generally aren't in your blood, they're in the tissue that got infected. And so your white blood cells have to get there, cross the membrane, and get into the tissue to do any good. Excessive amounts of this damages the membrane. I mean, blood cells forcing their way between epithelial cells causes those epithelial cells to be disrupted. So cellular injury, water comes in, because sodium-potassium pumps aren't working. Therefore, you build up intracellular sodium. It pulls water in by osmosis. But other things come in and go out too. Damaged cells become leaky cells. If you have damaged or dying heart muscle cells, they will leak some of their own specific troponin into the bloodstream, and you can measure it and tell if the person's had a heart attack. So the cell membranes become leaky. Things come in that aren't supposed to. Things go out that aren't supposed to. Things that were supposed to be inside the lysosome or inside a mitochondria or something leak out into the cytosol. And that adds even more osmotic force inside the goo of your cell to draw more water in and cause the cell to rupture. So here we have an injury resulting in hypoxia. You could even cut yourself, and what you've done partly is cut some blood vessels that might be supplying some cells right there. So maybe you didn't even cut those cells. The cells you cut are dead, but the ones right next to it might become hypoxic. ATP production decreases, sodium and water move into the cell, osmotic pressure increases, you suck water in, the cell starts to swell up. The first thing it does is create vacuoles, like water bubbles inside the cell, that if you can solve the problem quickly, the cell might be able to get rid of this excess water and survive. But if it doesn't, it will cause damage to the structures inside, cause the cell to die, or even cause the cell to rupture. Hydropic degeneration means water-based degeneration. And so cell death happens in a couple of ways. The cell can simply become so damaged inside it stops functioning. It can swell and burst. The nucleus, which is like a cell within a cell because it has a membrane too, can swell and break apart or burst. The term karyo means nucleus. So karyolysis is the nucleus just kind of disintegrating. Now the cell can't function anymore, it's dead. Or karyorexis, swells up and pops. Rexis means to rupture or burst. So karyorexis, this nuclear membrane has been stretched to the point where it actually pops. Necrosis is the term for dying, dead cells. So necrosis, if you have necrotic tissue, it means that you have cells in there, a group of cells has died. Coagulative necrosis happens in kidney disease, ischemia, where the cells die in place, they're dead, they change color, they stop working, but they still look like cells. They just look like dead cells. Liquefactive necrosis usually happens when you have bacterial infections and they actually eat the cells and cause them to turn to mush. Somebody with, and this happens in the brain with brain infections, brain diseases, and they say the tissue in there was like wet toilet paper. So the hydrolytic enzymes break down the cells and the bacterial infection starts breaking down the cells. Staph infections, strep infections, E. coli will tend to produce liquefactive necrosis. Caseous necrosis is sort of a combination of the two. So the cells die and then they're just really destroyed by a variety of enzymes and maybe bacterial action. This happens in tuberculosis, in the inside of the tubercle, where the bacteria are active, and you develop this cottage cheese effect. It's still a substance, but you can't see any cell structures anymore. It's too disrupted. But it didn't turn into a liquid. Fat necrosis happens when the enzymes inside the fat cells themselves or other enzymes produced by the tissues around them start damaging the fat cells. Pancreatic disease is a good example of this because the pancreas creates pancreatic lipase. Lipase is designed to digest fat. If the pancreas is blocked and is producing lipases and the lipases build up inside the pancreas, they start dissolving or eating up the fat cells that are in there. They damage the internal structures and turn those triglycerides into soaps. They actually react with the triglycerides inside the fat cell, and it kills the fat cells. Gangrenous necrosis is related to anoxia. Generally speaking, although it's not entirely anoxia, for instance, frostbite, it kills the cells through freezing them and causing ice crystals to develop inside the cells, but it also kills the blood supply to the cells around there. It allows a bacterium to build up inside the tissue, and it tends to spread and spread and spread. If you don't catch it really soon, you have to amputate toes, feet, part of your intestine. Here we have a thrombus that has blocked off blood flow going to the large intestine, causing a necrotic bowel. If this actually dies, you have to go in there and cut it here and here and attach the good pieces together. Strangulated hernia. If you get a bad inguinal hernia and part of the intestine sticks out, it can become pinched off, strangulated. It can't get blood flow out here, and this part of the small intestine will die. Again, you're going to have to cut it and attach the right pieces together. A volvulus is a twisting of the intestine. It can happen to the colon or to the small intestine, blocking off the blood flow. Cut it off, reattach it. Intussusception happens with children and fetuses, especially during fetal development. Part of the intestine slips inside the other part. It's called telescoping. And these creases here pinch off blood flow, and you get parts of the intestine here that start dying. And again, frostbite here, gangrene. Here is gas gangrene. This is the kind that soldiers in conflict used to get anytime they got shot or cut. We didn't have a lot of people coming home from World War II, with the kind of injuries that we have now from Afghanistan. Because back then they didn't have antibiotics. And if you got, gangrene, they either cut your leg off right away or you died. So, all the Civil War guys that survived their injuries all were missing an arm or a leg. Now you come home with a variety of horrible injuries, but they managed to solve them. And so we actually have a lot more walking wounded now than we did back then. But Clostridium perfringens is the bacteria. Again, this is one of those bacteria that's always on your skin, kind of like MRSA or staph. But your body controls it unless something goes wrong, and then suddenly it's opportunistic. It jumps in and starts eating away, and it produces, these are called bulla, B-U-L-L-A. They're balls of either fluid or gas. This guy had his leg completely cut open, and this is all liquefying tissue. This is liquefaction necrosis here. It's being eaten by the bacteria. Once they cleaned it up, it was all bright and happy. This is actually an okay leg. This leg survived. What happens after they open it and clean it, it is able to, you're putting the good cells next to good cells instead of being separated by goo full of bacteria, and so they can start growing new structures. The first thing they do is grow something called granulation tissue. It's a very fast-growing metaplasia type cell that provides a scaffolding for normal cells then to grow in and fix the wound. So here's your summary slide. A lot of people love these slides. Necrosis types: coagulative, liquefactive, caseous, fat necrosis, and gangrenous necrosis. Your types of necrotic tissue. Now, when cells die, there are really two reasons a cell will die. One is it wants to, and the other is it's killed by something. Apoptosis is a process that all of your cells have within them. You have what's called a suicide gene that is activated if the cell becomes so damaged that it can't function and can't recover. So you activate the suicide gene, you actually break apart the internal structures, you leak out the substances from there, producing a situation where water can flow in and the cell can be broken apart, the nucleus breaks apart, you know, it's drawn in by osmosis, it breaks apart, and the cell kills itself so the healthy cell next door can divide, and now you have good cells again. And this happens millions and millions of times a day. When our cells become too aged or too damaged to do their job, they will kill themselves and allow themselves to be replaced with a healthy cell. Necrosis is when it's killed and doesn't want to be killed. It's starved for oxygen, it's killed with chemicals, it's killed by a bacterium. So necrosis and apoptosis, you both wind up with a dead cell, but one is it was intentional and the other is not. And here are the macrophages. At the time the apoptotic cell dies, it gives off signals for local macrophages to come and eat it to clear the area out. So it's a nice, neat process of not only killing a damaged cell, but getting rid of it, so there's room for the new cell. Aging versus disease. All right, if you were to look at me and say I'm 18 years old, I would be diseased. You know, I have some wrinkled skin. I have a heart that has probably a certain amount of vascular damage, because everybody accumulates vascular damage over the course of their life. My brain might have atrophied some compared to when I was 18. I'm smarter now because I hadn't learned as much back then, and I was 18. I mean, how smart can you be? Sorry if you're 18. But aging versus disease, at age 66, I'm not diseased at all. I'm what I'm supposed to be at 66. So there's a certain amount of degeneration that is expected, and that's called aging. If you exceed that level, though, you have disease. If I had 4 blocked coronary arteries, I would have heart disease. That wouldn't be just aging, it would be heart disease. Normal lifespan is how long you should live at the time you're born. I was born in 1951. My lifespan at that point would have been about 73 to 75. That's about what it was. In the 1900, when Americans were born, their lifespan was 47. We didn't have antibiotics, we didn't have vaccinations, and people died. Now, people didn't get to die of old age. They always died of something else. I mean, 47 is pretty young. So that's what your life expectancy, or normal lifespan. If a kid is born now, they're a male, I think the normal lifespan is almost 80. It's increased quite a bit from 1951, largely because of medical advances. Your life expectancy is how long you're expected to live from this moment. So at age 66, even though it was 73 when I was born, now that I've made it to 66 without dying, and I'm pretty healthy, my life expectancy is probably 85. So it keeps pushing. My mother's 96. You know, when she was born in 1920, her lifespan expectancy, her normal lifespan was about 73 for a woman. You know, but now that she's 96, it's certainly not 73 anymore. She's healthy. She probably still has another life, you know, I mean, life expectancy is still another 3 or 4 years, or 5 maybe. So life expectancy based on your current age. With aging does become cellular changes. Aging produces a lot more fibrosis, adhesions. You lose your flexibility. You have more fibrotic tissue and less of the soft, muscular tissue that you had when you were younger. Accumulation of injurious events is one theory for why that happens. In other words, I have bumped my arms thousands of times in my life, just knocking into things, and the recovery process has always produced a little tiny bits of scar tissue, and over time, my heart has beat so many times, it has actually worn itself out through just repetitive damage. That's one thing. The other is it's genetically programmed. We know that people just can't live beyond about 120 no matter what. Now, of course, you know, part of it could be that they're beating themselves up over 120 years. But the other is we have, and we'll see as we talk about some more genetic, you know, abnormalities, telomeres on the end of our DNA that when they break off, you can't reproduce those cells anymore. So there is a programmed life expectancy as well. But this is a multifactorial thing. Both of my parents lived into their 90s. All of my aunts and uncles have. So I could expect to live to be 93. Will I? I don't know. Depends on if I get hit by a truck. Depends on how much damage I did to myself when I was a rock and roll star. You know, it depends. So it's obviously a lifestyle choice thing as well as genetics will decide how long you live. Atrophy of muscle with aging, decreased function of a lot of tissues. The GI tract does not produce quite as many digestive enzymes. Your liver doesn't produce bile quite as well. Your thirst mechanism doesn't work as well, so you tend to become dehydrated because you're not thirsty enough to drink as much. You know, a lot of things are simply a degeneration of tissue and a loss of function. Sarcopenia tends to happen in older people. A muscle decrease. Their muscles get so tiny that they fall over and break their hip. And the hip has gotten osteoporotic, or at least it's weaker than it was when you were young, so it's easier to break. A lot of this can be delayed. You know, there are 90-year-old people running marathons. There are 90-year-old people lifting weights and competing in the Senior Olympics. Not a whole lot of them, but my mother's 96 and she walks a couple miles a day and she does line dancing and her muscles aren't that bad. And they're a lot weaker than they were when she was 60, but they're still pretty good. You know, she's not moving around with a walker. Frailty is the term for when the degeneration of these tissues becomes to the point where you really cannot do what you want to do or need to do anymore. And ultimately, you die. Death of the entire person is somatic death, meaning body death. Some of the changes that happen with death, of course, you stop breathing, your heart stops beating, and therefore you stop producing, delivering oxygen. You stop making any ATPs anymore. Algor mortis, loss of temperature. If you can't split ATP, you're not producing the heat that keeps your body temperature up. Algor mortis, loss of temperature. Livor mortis, blood pooling by gravity. And that's, if somebody lies in one position for a while, you turn them over and they're purple on the bottom. Rigor mortis, ATP loss, doesn't allow the ATPs to bind and release cross bridges that have bound, and eventually all your cross bridges wind up getting stuck and you're stiff for a while until the cross bridges just start dissolving away because you're dead. Autolysis, liquefaction of tissue, release of rigor mortis as the cross bridges start to dissolve. And on that happy note, I'll see you next class.